So next is Brian DePasquale. So he's from BU and Department of Bioengineering. And we had discussions about foundation models. But he's going to talk to us about foundation model of odor system. So that's maybe this sort of approach would be interesting, having like a more specific sort of foundation model rather than general foundation model.
Got it. Yeah, I don't think.
This is it.
Yeah, you just plug it into it. And then you should have it's kind of worthy.
Okay. Hi, I'm Brian DePasquale. I'm from Boston University. I'm an assistant professor in biomedical engineering, and thanks, Nima, for having me. It's really fun to be here. So I started my lab about 2 years ago, and so I'm going to talk to you a little bit today. My title is Learning the Language of Smell, Foundation Models for Protein-Odor Interactions. So I'll spend a good chunk of time talking about some new work in my lab focusing on this space, but I'm also going to describe a few other projects that are a little kind of closer to work that I've done in the past. So it's a little bit of like a kind of distribution of the budding things going on in my new group. So I was thinking about the concept of open science, and it's really great to be here. It's really interesting, but I don't really think of myself as like a huge kind of like leading the charge in open science. So I was kind of reflecting on the term open, and I kind of am going to reinterpret it for my own talk. I think typically when we think of open science, we're thinking about sharing data and making sure that data is accessible, this kind of things. I'm going to kind of invert it and suggest that, another way to view open science is to open up yourselves to new ideas. I'm going to introduce kind of two new ideas that I think are pretty interesting. I've been interested in them too. And hopefully you'll kind of be exposed to some new thoughts. So I'm really thinking about kind of opening your minds to some new thoughts. And then the last part of my talk, I'm going to talk about something that I've been talking about a few people with, which is some programming that I've been doing in the Julia language, which I know is sort of a lesser used language. So I'm going to implore you to open your hearts a little bit to the possibility of getting interested in the Julia language. So a little bit of an overview about what I'll talk about, depending on how much time I have. Some extensions of some older work that my group has been working on. We've been thinking about building some multitask neural networks that are composed of spiking neurons to think a little bit about the origins of spiking variability. We've been developing some hierarchical models for strategy switching, and then, as I said, the title of my talk is on foundation models for olfaction, with a little bit of a kind of a quick side quest for graph neural networks, which is a topic I've become very interested in, and I'm also very interested in proselytizing to this community because most people don't seem to know about them or use them, and then again about an open source code base that we're developing. This picture is pretty common. I think we're all kind of on board with it, that a really great way to think about neural responses is as some kind of low-dimensional activity in some kind of manifold. And this has become now kind of one of the most widely used abstractions in neuroscience. A few years ago, Mark Churchland and I kind of set out to think about how we could link up this picture to maybe a more mechanistic model description of how a neural circuit is actually constructing these low-dimensional manifolds. And the reason we thought this was important is because typically this description was being derived from the kind of view of data analysis opposed to a mechanistic biophysical model of how you would construct these things. I'm not going to talk about the method that we developed for building these models, but basic idea is that we wanted to kind of invert this relationship between these latent factors and spike trains. So they typically derive from this notion of latent factors kind of give rise to some probabilistic activity of a neuron. We wanted to kind of think about these mechanistic spikes as being kind of the way in which you construct these low-dimensional trajectories. And then we wanted to use these to kind of relate these statistical models to kind of more mechanistic models. So again, I'm not going to tell you how we built these models, but we wanted to construct them to generate some data. So we took some principal component trajectories from data from his lab, monkeys performing a cycling task. We derived some PCs from them. And then we were able to successfully kind of embed these principal components in a completely deterministic neural network, spiking neural network model that was able to construct the cycling EMG from that model. And then once we built these models, we realized we could kind of take a look inside and understand how they were working. And one kind of key insight that we derived based on the model structure is that we were able to kind of get a better picture into the relationship between the firing rate of a neuron and its kind of trial-to-trial variability. So what I'm showing you here is the input into a neuron in this model, and it's being separated into these kind of two components that we put into it. One is this kind of reliable input, which is due to these low-dimensional trajectories that we train the model to produce. And the second part of the input is coming from a random synaptic component that we built into the model and that we didn't alter. And so if you look at the activity of this neuron as being the sort of sum of these two parts, it's essentially producing reliable output from trial to trial due to this reliable input, which is essentially the projection of the low-dimensional manifold, and some unreliable activity that was due to another kind of unquenched synaptic input. So we were able to, in that paper, build these models so that they could do more than one task. This is kind of ongoing work that we're interested in moving into. So the idea we had was, what if we take these models and train them to construct multiple manifolds that are orthogonal to each other, and these manifolds are each doing different tasks. So imagine a scenario where I'm training a model just like I described, where you have a manifold doing one task, a manifold doing another task. So the one that we did in our paper was we constructed a network to do a reaching task, sort of a standard Churchlandian reaching task, and then we built a model to do both tasks. So what I'm showing you here is a model doing the cycling task, and we constructed it so that the activity lives in these sort of cycling directions when it's performing cycling, and in the reaching directions it's not doing anything. And then when the system performs a reaching task, in the reaching projections it's very active, and in the cycling directions it's not doing anything. And so we were able to kind of switch back and forth between these two different tasks in a way that we thought was pretty interesting. So where we're going with this is, I really like to build these models to try to introduce a hypothesis about what the origin of spiking noise might be. Historically, the origin of spiking variability has typically been justified or thought of as being produced from synaptic balance or really tight synaptic balance. I don't believe this hypothesis has been considered before, but it might just be due to essentially building a model that does lots of stuff, and then you start to impact some kind of constraint on how many tasks you can do. So the idea I'm pursuing in my lab right now is that when you're performing, let's say, one task, activity kind of resides in this low-dimensional cycling space, and provided you're kind of executing that task correctly, you'll kind of reside in that space only, and then due to small fluctuations in the input, you might kind of jump off the manifold there incorrectly, moving into one of these other orthogonal directions, and that's going to lead to synaptic input coming back into these neurons, which would produce irregular spiking. So I haven't yet started really building these models yet, but I think it's an interesting use case for them after we essentially built the technology to construct them. And one way you can kind of conceive of what's going on here is it's very similar to the kind of capacity limits of a Hopfield network. The more memories you pack into a Hopfield network, you eventually hit a capacity constraint, and there's a degradation in memory. Spiking noise essentially is the same manifestation of that for doing more than one task. So that took longer than I thought it would, so I'm going to jump over to what I was originally going to plan to talk about, which is foundation models for olfaction. We're also, as I said, working on some hierarchical models for strategy switching. This is derived from work that I did during my postdoc in decision making. I'll just kind of breeze through these slides quickly to get onto the foundation model stuff, but the punchline is where a typical model that you might build for doing decision making is a drift diffusion model. We know that animals don't do the same thing from trial to trial. So we're building some models where we stick an HMM underneath it. We're calling them HMM DDMs. And we can now essentially model trial to trial fluctuations in decision-making strategy that animals exhibit. This is in collaboration with Ben Scott, who's at BU, and with my graduate student, Ryan Sen. And these models work pretty well. Turns out, no surprise. Making them state-dependent, you do a better job. Okay, so I want to talk now about a little bit of work that we've been doing in foundation models for olfaction. As I said, this is kind of like a new direction for my group. It seemed like a really great chance when I started my lab to do something pretty different. We'll talk a little bit about graph neural networks on the side because they're relevant to these models and I think also very interesting for this community. So the motivation really was the huge success story of vision. I think like as a community, we've done a really, really great job at modeling the visual system. And analyzing the visual system, it's a very impressive body of work that I feel like pretty jealous of, actually. I think the real success there has been these kind of analyses like coming out of Dan Yeaman's group and Jim DeCarlo's group, where they build these kind of simultaneous convolutional neural networks alongside real data from the visual system, and they're able to accurately predict neural responses like relatively deep into the into the visual hierarchy with those models. There's no, this should be just, not really a surprise to anyone. We've done a really great job in the machine learning community as continuing to develop these models. This is the performance on ImageNet as a function of time over the past few years, and we're essentially hit human level performance. So I was kind of looking for something that we weren't doing a very good job in, and I think there's a really clear case to be made that we don't really do a very good job in olfaction. And I think the clearest, all you need to do is like kind of look at our best chemical sensing robot, and it's still like not, it's a dog basically. Like these are the best robots we can develop for chemical sensing, which is a little surprising. I mean, every organism on this planet has a chemical sensing apparatus, and it feels like we should probably be able to make a little bit more progress on this. So that's kind of like, back to my idea of open science, open minds. What I'm going to tell you a little bit about is some of these foundation models that I've been exploring in chemistry and protein biology, because I think that they are really applicable to olfaction. I think there's probably going to be some interesting use cases in other spaces in neuroscience. And I think there's probably some lessons to learn about foundation models broadly that we might be able to adapt to neuroscience in some way. And so the punchline a little bit is that there are some really great foundation models in biochemistry that seem right for olfaction and neuroscience more generally. So just a quick refresher, if you haven't thought about smell for a little while, you have olfactory receptor neurons in your olfactory epithelium. They have olfactory receptor genes in them. These olfactory sensory neurons synapse into olfactory glomeruli. Depending on the species, it's all pretty segregated. It's not exclusively true, depending on what species you're looking at. And these neurons then send their projections deeper into the brain that give you a sense of smell. Obviously, the olfactory system is a little bit more complicated than that, but I'm going to actually focus on the most extreme part of it. There's the olfactory periphery, which I think there's a lot of heavy lifting that the olfactory system is doing. In some ways, it's actually quite different than like what we might think from our models of vision. So, I mean, that's basically the problem of olfaction. You have a lot of odors, you have a lot of receptors, and you have to figure out who connects up to who. I mean, in some ways, it's basically that simple. So what's the problem? I think there's basically kind of two problems from a machine learning perspective. One is that chemicals are kind of oddly shaped. This kind of stands in contrast to olfaction. We basically are dealing with squares. Obviously, machine learning is pretty good at dealing with squares. How do you deal with oddly shaped compounds? That's where I'll start talking about graph neural networks, which I think has been a really handy tool for oddly shaped things. And the second problem is really kind of a problem of data. So depending on the species, you have roughly 100 to 1,000 olfactory receptors. There's not great estimates on how many compounds exist that have a smell. There's, maybe 10 to the 5 seems like an approximation. Someone postulated 10 to the 9 a few years ago. I think it was pretty roundly rejected as being too large. But I think, it's an open question how many chemicals have a smell, which I think is an interesting problem. The data sets you can find are roughly on the order of 100 to 1,000. So we're kind of in a super low data regime here. We don't have a lot of data, which means that foundation models can perhaps be helpful. We can leverage other knowledge we have about chemistry and biology to help us in this scenario, but we're not going to really be able to build large models with these data directly. So the first question I brought up was odd shapes, and so this is where graph neural networks come in. This was a project that a student really kind of drove, he kind of really spearheaded. He had a background in computational chemistry, and he wanted to get interested in neuroscience, and I said, hey, maybe this is like olfaction, maybe seems like the right thing for you. So for those of you who don't really know how to adapt chemicals to kind of graph neural networks, so here's a chemical structure. It's basically a graph. So you kind of featurize it in a way that you would featurize most other kinds of data. So each atom, you would featurize it as a node, and you'd give it certain properties. Its atomic charge, its valence, is it part of an aromatic ring? You'd basically give each node all of the properties of that atom. And then you describe the connections between nodes as edges. In a graph neural network, you pass them those feature properties through a deep neural network to adapt them into a different form so that you can do some kind of prediction task. In this case, what I'm going to describe is we're trying to predict neural responses from the properties of chemicals themselves. So a quick aside, just because we're sort of hot on the tails of it, I had a workshop at Cosine I organized with Kim Stockenfeld and Sam Llewellyn on graph neural networks. If you're interested, there'll be a QR code at the end. We tried to aggregate as much information from that as possible, And we had a nice tutorial for those interested in PyTorch applying graph neural networks to connectomics data from the Flywire data set. So if you're interested in Flywire based on the previous talk and interested in graph neural networks, we had a nice workshop there. I'm going to breeze through this a little bit. This is kind of like a little bit of a, like, why the heck isn't everyone doing this? There's so much geometrically complex data in biology. Here's 8 examples. I think that graph neural networks seem like the right hammer for the right nail. There's a lot of nails out there, so if you're interested, happy to talk about it. Also, you know, a little bit of kind of like, what the heck are we doing in neuroscience question. This was from the workshop. I computed the frequency of the occurrence of these two terms in both PubMed and in cosine abstracts over the last 10 years. Graph neural networks was reflected 5 times. 2 of those were me. And last year, cosine recurrent neural networks was used 86 times. So we perhaps maybe need a little bit of openness in what models we're thinking about using. I think graph neural networks are pretty ripe for using it in neuroscience more broadly. Other fields in biology, as represented by the PubMed citations, are using them quite a bit. So I want to tell you quickly about 2 foundation models that we've been using. One is this model called ESM. For those of you who don't know, these are called protein language models. You essentially take a transformer and you give it a humongous data set of amino acid sequences. You occlude a small number of residues from the sequence and try to predict the missing residue. So this was work that came out of Facebook. This Alex Rivas now is at MIT, I believe. He just moved to the Broad Institute. And what's really interesting about these models is that you can predict quite a bit from them after you train them. So this is trained on around 250 million proteins, and inter-residue distance can be predicted afterwards directly from the model, which is what I'm showing you down here. Needless to say, there's a few other tasks that they did, and I can't walk you through all these tasks here, but this is from their paper, and they show that pretty much any kind of functional assay that you would want to try to understand from a protein based on how much the protein mutated, their model was kind of blowing everyone out of the water. So these are surprisingly under-noticed in neuroscience, given that we study an organ, a biological organ. They're, I think, really kind of running the show in biology outside of neuroscience, and I hope that others kind of become more curious about them. A separate class of models we become interested in also are foundation models for chemistry. This is a model called MoleClear, where they are essentially doing contrastive learning, which is a technique was described earlier, I think yesterday, for images, where you occlude some aspects of the compound. Up here you can see occluding nodes in a chemical, or bonds, or subgraphs within a compound, and trying to predict those missing nodes. This model, I believe, is trained on 75 million unlabeled chemicals, and they were also able to show that this kind of foundation models are really great at predicting chemical structure afterwards. This is a second model called ChemBERT, which I don't have the time to describe, but it's a large language model adaptation of BERT towards SMILE strings, which are a sort of character sequence strings for chemicals. So I have about 2 minutes left, and I'm going to finally get to something that I've been doing. The questions we really were focused on were, can we use these foundation models to predict odor receptor binding? And we were initially kind of curious if chemicals alone would be enough to describe these neural responses. We were interested in seeing if we could include protein information, and if that allowed us to do better prediction. And there's a lot of different ways, there's quite a large number of foundation models in chemistry. We were curious to kind of assay them and see which ones were performing better than others. The models we were typically using were to take the chemicals that we were studying, put them through one of these foundation chemical models or foundation protein models that were pre-trained on some kind of task, and then try to predict which compounds were going to bind to which proteins. We tried a few different kind of decoders from the embeddings. We tried a linear-only decoder from the molecules only. We tried a linear-only decoder from molecules and proteins, and then a neural network trained on the bottom half of the embedding on both proteins and molecules. These are the 2 data sets we use. This is a pretty typical classic data set for olfaction from John Carlson's lab. This is probably one of the largest data sets that exist, and it's only about 100 chemicals for about 24 olfactory receptors. Not a lot of data. This is another data set that we were using, this M2OR data set. This was the largest data set that I could find, about 53,000 pairs of odors and compounds that bind. And to my knowledge, this is the largest olfactory data set that exists. The first question we ask is, how well can you do if you just know the chemical identity? The answer is, surprisingly, is basically you can't predict anything. And I was really surprised that this had never actually, I cannot find this in the literature anywhere. If you train a neural network model on various forms of chemical embeddings, you essentially get zero test performance. So chemical information alone is not sufficient to predict a neural response on an unseen data set. And that was true for both the two different data sets. What I'm showing you here at the bottom is the various types of chemical foundation models we use. I don't really have time to go through them in the interest of time, but we tried a bunch of them and none of them seem to really work very well. The interesting thing is if you add the protein information from a protein foundation model, you start to do quite a bit better. So what I'm showing you here is R squared for the Hallam-Carlson data set when you include protein information. And now we're doing pretty good, about 0.4 kind of across the board. Same for the M2OR data set, a little bit better. The good, and if you make the decoding that you're doing from the embeddings a little bit more complicated with the neural network, then you do a little bit better than that. The last thing I want to note is that one thing we were really surprised by is that the chemical foundation model basically doesn't seem to do anything. You're not doing any better if you embed your data into a foundation model than if you just use what are called physicochemical descriptors, which are basically properties of the compound itself. So I think that the real kind of intuition I've gleaned from this is that there's quite a bit of space left in pre-training chemical foundation models for improving them. I think they essentially lack the kind of context that we know is very effective for other types of models. So that's the punchline on foundation models for chemistry. Can you do this with chemicals? Not really. If you add protein information, how do you do? Quite a bit better. And chemical foundation models, I think, have a little bit left, of an improvement for, at least for predicting olfaction. I'm out of time, so I just want to jump to the last bit in about 2 minutes, which is maybe to consider something that you haven't maybe considered in a little while, which is that Julia is a really fantastic programming language that we maybe should all be using a little bit more. And I don't want to like open up a Julia versus Python debate. I think that's the wrong debate. People in my lab use Python all the time. But I think Julia is also a really great language that we should all maybe think about using a little bit more. I really like this meme. I think it's like kind of like my own strong view on Python. This is from Miles Craner. He's, I think, a pretty strong Julia enthusiast. He says, the more I use Julia, the more Python and its numeric libraries look like a Victorian-era stagecoach with jet engines duct taped to it, each pointing in different direction. And that's exactly my own view of Python environment. I think Torch is great. I think JAX is great. I think NumPy is great. They're all really powerful, but they're kind of attached to something that was built, almost 30 years ago. So I don't know if that's maybe always the right thing we should be driving around. I think Julia does a great job in ways that that Python also does a great job in. I think what I want to say just quickly is that one of the things that is the real selling point for Julia for me is the ecosystem is really easy to interface the different libraries that you're using. So it's really kind of considered a library of libraries in a way that I think Python really struggles with because of that kind of buggy stagecoach at the base of it. I'm happy to talk more about Julia after my talk. But I just want to do a quick advert for a package that we've been developing called State Space Dynamics. This is like 100% inspired and thanks to all the developments that Scott Linderman's group is doing for a long time in state space modeling. We wanted to kind of introduce some of those that similar functionality into the Julia ecosystem. So it's very similar to that flavor. We're thinking mostly like the kind of state space models that have been really successful in neuroscience over the last few years. We've been focusing on hidden Markov models and linear dynamical systems, and trying to make it pretty fast and pretty user-friendly with documentation and stuff like that. Here's a simple example of a Poisson linear dynamical system that's working pretty well. So the functionality is getting built up slowly. We are, not sort of at Linderman level status quite yet, and I doubt we'll ever get there, but that's a different story altogether. But if you're Julia curious, I hope this is kind of like an entry point into it. The QR code is here. I'll get to it in a second, but the QR code's there if you want to take a snap and you're curious about Julia. We're always looking for new contributors. And so the punchlines really for me are, for my talk, is that there's, I think, a lot of really interesting foundation model work going on in biochemistry. If you're curious about it, I'd say take a look. I think there's some lessons for neuroscience there, and also some applications to neuroscience. And if you haven't taken a peek at Julie in a little while, maybe take another peek. I think there's some nice progress that's been made in that ecosystem. So with that, I'll thank my collaborators from the spiking neural network stuff, my postdoctoral advisors for some work that I didn't really talk about. And I have my new-ish lab at BU. We've been there for about 2 years. Grant and Ryan were mostly kind of spearheading some of the projects I talked about, and I'm happy to take any questions.
Scott.
Yeah. Oh, yeah, for sure.
I know shares a lot of your enthusiasm for graph neural networks and olfactory responses. And many of us know him for his work on MoSeq,
Yeah.
the data lab, but the reason he actually joined in the first place was because their olfactory side of neuroscience research they were doing.
Yeah.
Anyway, they have this amazing stuff on like principal odor maps using GNNs, and they trained all these panelists to distinguish various scents. It's really cool stuff, and it seems like it would be super useful for olfactory neuroscience as well. And I'm curious if that was one of the things you've played with in those.
Oh, yeah, no, definitely. I mean, I read his work like not long after I started my lab at BU, and I was like, oh, this is the way to do this, for sure. And then my student who had a background in chemistry was like getting very curious in graph neural networks also. I think that work is really interesting and great. I think I think there's a few nuances that I think there's some more interesting work to do. I mean, they're training it to human perceptual data. I don't think that, and I know they've explored the space of olfactory receptor activity also. I think there's a lot of interesting work to do there. Those data sets tend to be quite a bit tinier, so there's some more work there. And to my knowledge, at this point, I'm not sure that they've started using the information from pre-trained models in those models. Those are essentially straight up self-supervised learning. There's no pre-training going on there. You'd have to imagine, I mean, he's a really smart guy. I'm sure he has his ear to the ground. I'm sure he's trying it, because why wouldn't you? I think it's an open question. In our hands right now, for example, those chemical foundation models aren't doing anything for olfaction. It's not clear if olfaction's just like a different problem than whatever kinds of problems people have been working on biochemistry. Or I think in the community of people pre-training chemical foundation models, it's not really clear what context is in the context for a chemical. So I'm not sure they have the pre-training quite right. But I think it's a really interesting question how you could get it right.
Really great talk. Another really interesting point of contact is I know one person in WashU really interested in just stuffs, Timothy Holy. And I think his approach or idea is kind of also related but different. Since the binding problem is kind of biophysical problem, should we take into account the structural informations instead of working in the latent space? Instead of latent space similarity, should we just think about the 3D morphology of the chemicals and the things, and those may be more informative to the fundamental processes of binding instead of the latent space, which we kind of don't quite know how they works. Have you any thoughts on that?
Yeah, I think that's a great question. I mean, I have some collaborators who work in kind of more old school, physical simulations of binding site locations, and I think that there's probably an interesting opportunity to interface those two. So using some of the more kind of generic foundation models and embedding space models for doing this prediction, augmented with some knowledge about those kind of physical, simulation models. But I haven't worked in this space long enough to become an expert in any sense. But yes, I think, we have part of this work stemmed from the fact that I was in a biomedical engineering department, mostly with people who are doing this kind of work, and I was like, what the hell am I going to talk about them all with? So I started to kind of migrate into molecular biology a little bit more. But I think, I think there's probably some interesting opportunities to interface those two kinds of two types of approaches.